As good evening once again to all the participants. So, welcome to session 13 on tools and databases for computational vaccine design. So, before going to this, if we want to work on the other tools, okay, like whatever we have seen earlier in the previous session. Okay, in the previous session, we discussed on B cell epitope prediction. Okay, and for talking the peptide protein, like whatever epitopes we have predicted, if you want to talk with the toll-like receptors for DNA and RNA viruses or even for the bacteria, we have to download the TLR proteins from website. If the structures are not available, model it, and then we have to talk with the online servers. One such online server is CABS Talk. Okay, so in CABS Talk, we can upload the PDB file and peptide sequence, and we can make it. And I was explaining you on the last session about the prediction of properties from the structure, isn't it? From the whatever peptide we have predicted, okay, the structure is predicted, we can also measure the properties. Okay, for that we have to go to the B cell epitope from IEDB. So we can see the tools, plenty of tools there. Even if we want to check specifically on, now I take you to the B cell epitope. In B cell epitope, for predicting the structure based epitopes, we have gone to this photo. For linear based epitopes, we have gone here. Okay, and this is epitope prediction based on structural protrusion. Methods for modeling, we can use this. And for peptide based, I think we can able to find in Raghava web server. You remember in Raghava web server, we have some tools even for predicting the peptides. Okay, so I think we can better take to the same, the previous web server uh, in Raghava toxin pred. So for predicting the toxicity of the peptides, we can go here. For designing peptide, we have information. For batch submission, we have that information. Paste the peptide, and we can run analysis. Okay, virtual screening of toxic peptides can be done. Like you have to paste the multiple peptide sequences. Okay, if you have screened some 10 peptides, paste one by one here. Okay, the paste only the peptide sequence uh, in FASTA format. Okay, or just paste the sequence. And after that, what we do here is we are going to check which is toxic and which is non-toxic. Okay, so for this we can able to do the virtual screening here. Okay, so this is what the toxicity prediction through Raghava web server. And the immunogenicity for T cells can be predicted from the other tools. You see immunogenicity, it's not directing take to the immunogenicity, I take you to the T cell epitope. If I click the top one T cell epitope, so other than predicting the T cell epitopes of MSC1 and MSC2, we can also see the immunogenicity prediction, like how we find a toxicity prediction separately from Raghava web server. Even immunogenicity tool is available. Okay, we have proteasomal cleavage separately, cleavage site separately, and for immunogenicity predictor, this is the tool. Okay, like this, we have plenty of tools for testing the structural complex of epitope receptor. We have this tool for talking of the peptide based. Okay, you can go with doctor. This tool is available in IADB itself. Okay, so like this, we have plenty of tools in this T cell epitope prediction tools. Okay, now I think I have completed the tools available in IEDB for predicting immunogenicity, toxicity from Raghava web server, and what is the tool we can able to use it for peptide protein talking. Okay, now let me take you to this session. Okay, so I just recap what we discussed in the previous session. Now we can go to session 13. So in session 13, we majorly discuss on vaccine databases, vaccine tools, and the types of vaccines for prevention of diseases. So if we see for those who are working on veterinary vaccines, okay, and human vaccines, we have a web server like Human Vax from Violinet. So we have to go to this URL and we have to search for human vaccines. Okay, like for bacteria, virus, for all the types of pathogens. Okay, few vaccines data are deposited here, and next to this few vax, so what happened? The system got hacked. So now let me take you to the next screen. Licensed veterinary vaccines. We have to go to this URL. So here we have to select the animal, and then we have to select the pathogen, and then you can check the types of vaccines for these animals, and protein for protective antigens from Violinet. Okay, so type a keyword and you can search for it. And for DNA vaccine, we can go with DNA Vax TB database. So we can search through DNA vaccine query, plasmids, antigens, like for a DNA vector based vaccines. Okay, especially for SARS-CoV-2. Okay, there is a zoonotic pathogen, isn't it? SARS-CoV-2, Nipah virus, SARS-CoV-1. For all these kind of an antigens, we can go with this kind of DNA vax TB, where we can use even creating the DNA vaccines. Now, for especially for a DNA vector based, even for SARS-CoV-2, we have DNA vector based vaccines, isn't it? So that for vector design, we can go with VaxVec. This is another kind of a tool, so it can link with the databases here, and you can able to find appropriate kind of vectors, recombinant vaccine vectors, and we can design vaccines accordingly. And this is another database called Virulent Genes used for development of live attenuated vaccines. Virmugen DB, again from the same kind of server called violinet.org. So, if your own vaccine design and research, one has to go through this website. And Vaxor, that is for vaccine animal responses. For vaccine animal responses for different pathogens, we can able to find in this database. And Vaxism for vaccine mechanisms. For each and every, so generally we take a pathogen and we can take a subunit of a pathogen, or we can take a live an attenuated vac, or you can take a killed kind of viruses as vaccine antigens, isn't it? For a vaccine, I told you types of vaccines yesterday, that vaccines can be of live, attenuated, or killed vaccines, or even the toxoids produced from the bacteria, from the pathogens, can also be used as a vaccines. Okay, then we have to take the same pathogens for synthesis of vaccines, isn't it? So for this, we can use this kind of vaxism. And we have VBRC nerve tool. This can works on regional vaccinology, and these are the databases we can work with the regional vaccine design. So actually, the databases we link with this tool where we can work on this. And if you see this computational vaccine design, other than whatever I showed you in linear databases and tools associated with it. Okay, so that is coming under a kind of a vaccine design, of course. But initially, we have discussed on epitope prediction tools, isn't it? For epitope prediction tools, we have seen IADB, Raghava Web Server. Okay, so these are the servers we have seen. So this is one type of computational prediction. And the second is even we can predict the antibodies, okay, especially for treatment of cancer. The antibodies are the best therapeutics to treat. So it's for viruses we have to go with even peptides or with the multitopic peptides. What we call as multitopes can be used for targeting different pathogens, isn't it? But for cancer we have to model the antibodies. So one should have an antibody modeling kind of a software. Okay, if you see this antibody modeling, especially engineering of antibodies, I told you, if in an antibody we have a heavy chain, and to the heavy chain a light chains, I can take it in a different color. So the light chains are attached, okay, the light chains are attached in this antibody, isn't it? But one thing you have to remember, the most important in the antibody is this variable site regions, that is uh, uh, the paratopic region which will contact the epitopic region of the antigen is most important. So for designing this kind of this variable region, I mean in the variable fragment, so in the topmost I have a variable fragment. So that variable region, we call that as a variable SCFE fragments. We can engineer it. If we engineer, okay, in such a way that, okay, so I can take an eraser, I can erase this, I can erase this. So if I can even erase this line and I take this and I draw this. Okay, think like we have this kind of a antibody now. So in SCFE, okay, if I engineer in such a way that if my kind of interaction, for example, if I have these kind of structure, okay, and if my antigen, okay, is having, if my antigen is having such a kind of a fragment, SEF, if my antigen can be able to exactly bind in the binding site based on the engineering, earlier the fragment might be a state one, but I engineer in such a way that the structure of paratope can effectively bind with the epitope of an antigen, then you'll find you if you are treating for a cancer, obviously you can treat cancer effectively. So for precision targeting of cancer-like diseases, the structural modification of variable site region, SCFE fragment, what we call it as engineering of antibodies, is essential. Uh, okay, and so this can be done with this antibodyomics. So to work on antibiotics, we have a separate tools in Discovery Studio, Scroidinger. Only these commercial softwares have the antibody modeling tools. Okay, in the this is actually like a package. You can work with the antibiotics. Okay, but in freelance it's very difficult. We have to work with plenty of tools to come up with a conclusion whether the engineered antibody so, and the structure modeled after the engineering can bind effectively with the cancer antigen. So try with this kind of a commercial source. So for a Discovery Studio, we have an antibody modeler, and similarly, Scroidinger, there is a specific name for that for antibody modeling. If you make it, you can work on this computational facts. Okay, so this is for engineering antibodies. This is for simply modeling the antibody. And the fourth one is codon optimization. So you know that in codons we have exons, introns. Exon stands for coding proteins. So If only if you optimize it, optimize the codons, that is, if you go for exon, so the exons can also be retrieved from exomes. We have discussed on omics in the previous sessions that we have genomics, proteomics. So studying about genome is genomics, studying about proteome, proteomics. Like studying about exon is what exomics. And if you take an exome, okay, which can code for a protein, and if you work on a kind of a targeting, a protein targeting, then that's what we call it as the codon optimization tool is useful for that purpose. Okay, in computational vaccine design, the fifth part is the scaffolding. Even the scaffolding of proteins can interact well with the kind of an antigen protein. So, what kind of a scaffolds is generally used in the pathway, and we can. So, for making a scaffolds, we also have tools. For RNA scaffold, we have RNA scaffolder as the tool. For peptide scaffolder, we have PEP, and we have similarly Mammoth. These are the tools used for scaffolding. Okay, we have five different tools, five different areas in computational vaccine design, other than the peptide based vaccine design, which is the primary in reverse vaccinology in immunoinformatics. Even the other, whatever I mentioned, the four different strategies are also comes under immunoinformatics. Now, if you see this file, and this is the major home page, here you will find plenty of databases. So here you find plenty of databases to work. And even for the host responses, we have literatures, we have for vaccine design, we have separate tools. Okay, so tools and databases are interlinked to work on. Discoto, I told you earlier that based on the structure, if you feed the structure of a viral source, or even from the cancer source, we can able to predict the peptides. And this is another server called Vector Builder. In Vector Builder, for designing the DNA vectors, okay, we can use this kind of a software. Of course, this is also a commercial one to work. Types of vaccines and designs. Okay, so if you see for a multi-tool based, whatever computational vaccines, vaccines predicted from computational design. Okay, other than meningococcal polysaccharide, now we can see the next example is Kaposi sarcoma is predicted through immunoinformatics, that is reverse vaccinology. And for nosocomial infections of Pseudomonas aeruginosa, we can able to treat through multi-epitope vaccine. So this is done through comparative proteomic strategy. That is comparing proteome of Pseudomonas aeruginosa with the other Pseudomonas strains or strain proteomes. Okay, and then a DNA vaccine for Ebola virus. Okay, the strategy used is gene expression and immunogenicity. And similarly, for diseases gonorrhea, hemorrhagic, Watson cancer, Watts, the skin disease, okay, a viral kind of the virus can see such a kind of human papilloma viruses, okay, which can give such a kind of a skin disease. This can also be targeted through multi-epitope vaccine. So this is through structural vaccinology. So like this, see, we have so many kind of computational informatics, okay, who can use for targeting different types of disease. Other than the viruses, cervical cancer. For cervical cancer, we have peptide-based vaccine. So this is done through immunoinformatics approach. And similarly, for cancer, most of the types of cancer, we have dendritic cell-based vaccines. Our lab is working on the dendritic cell-based vaccines for more than uh, 18 years. And my mentor is working in this dendritic cell-based vaccines for 32 years. So, even, the Nobel laureate, uh, so the Nobel laureate who got Nobel Prize for dendritic cell, he received on 2017, Ralph Steinman, okay, from Rockford University. And similarly, can see for DNA vaccines, epitope-based, multi-to peptide-based vaccines we have developed from the immunoinformatics approach. And I told you in our lab, whatever is happening in the world, okay, so we are working on these kind of a vaccines in our laboratory. So majorly the recent type of adjuvants, instead of developing from the inorganic, now we are relying on the organic. So we take especially plant-based adjuvants like saponins from various plants or paste used as an adjuvants. Even for coronavirus vaccines, saponins are used as an adjuvants. Other than that, we can use the antibodies like interleukins and GM-CSF. Dendritic cells also used as a vaccines. So this is a database developed in our lab for vaccines. And for vaccines we can try this adjuvants, and we can check with so initially we want to check the efficacy of an adjuvants. So try to get the antigens without adjuvants, and we try without adjuvants, and after adding adjuvants to check what is the efficacy of vaccine antigens, okay, with and without the adjuvants. This we have tried, and we have checked with the dendritic cell, the different stages of dendritic cell through maturation markers. Then I also tried with T cell proliferation through T cell stimulation index. So we got positive results actually with these plant-based compounds. The plants I've chosen is the Borrelia diffusa. Borrelia diffusa is popularly called as Punarnava plant, and I also tried with ashwagandha plant and adjuvants. Yesterday also I mentioned we can take even the compounds like PEG for SARS-CoV-2 vaccines. We can take adenovirus. So adenovirus spike protein itself acts like an adjuvant, and the popular Sputnik Russian-based vaccine, they used adenoviral vectors as the adjuvants, and we have seen functional diversity of the adjuvants. Sometimes we use adjuvants in oil and water emulsion. Okay, we can use phytol-based adjuvants like phytonol, okay, as an adjuvant for increasing. So actually, there was we have to learn the immunology a lot to know about even the functions of these adjuvants. Okay, immunological action of adjuvants. In spike glycoprotein, there are plenty of mutations of available. In second wave mutations, we majorly found these kind of mutations happen in 1290 amino acid chain of the spike glycoprotein. So one mutation, this K lysine mutated to aspargine in 417. This is one strain, and K mutating to 309 in the same 417 position is another strain. Like this, I have hundreds of strains from SARS-CoV-2. So for each and every strain, we have to individually develop a vaccine. Not a single peptide-based vaccine can target all these strains. That also we should remember. And PEP43, I told you how to predict the peptide structure. This is used for predicting the properties. And patchdoc server, it generally asks a ligand. Now, initially, the previous session I told you for a PDB ID, if you download, okay, you can have a human antibody complex with the viral hemagglutinin. You try to separate these two different files, and whatever two different PDB files you have for antigen and antibody, load it in patchdoc and try to dock it. But if you try to dock it in the online server, we just get this information like score, area, AC. This is not significant, but one thing is significant, that is the result.pdb. So we had to download this result.pdb, okay. And also, other than feeding the ligand and receptor, one should also feed the receptor sites. I mean, what are the binding sites of the receptor? So if you know the binding sites of the receptor, feed the receptor site regions. These three inputs you have to feed to get this kind of a result1.pdb output file. So this you have to feed it in here. In which server? Firetalk server. In Firetalk server, again it asks for three different kind of files. Okay, so that PDB file you can able to feed as an input here, and you feed the receptor and ligand once again. Here only you can get an output. What output you can get? the global energy. Like however you talk without to talk, you get energy, isn't it? Similarly, we can get energy, global energy, attractive van der Waals energy. So these are the types of output we can get from protein talk. Okay, this we can able to show it. And in vaccination, one should also know some knowledge on the vaccination. That is, in morning vaccination, if we take that BCG, Bacillus calmette vaccine, okay, the interferon gamma, the interferon gamma is a very important protein, immune protein, for combating against different types of viruses. We have interferon like alpha, beta, gamma, but interferon gamma is more effective on combating viruses. And similarly, tumor necrosis factor alpha. Even in our tumor necrosis factor family, we have alpha, beta, gamma, and we have other family of proteins. But alpha, TNF alpha is the important protein for combating cancer. If this is for combating virus, this is for combating cancer. Okay, like this we have other side of kind interleukin called other interleukin called IL-1 beta. So one thing you have to remember, at 8 o'clock we find high cytokine secretion. But if the same vaccine, if they take at 6 p.m., we find lower cytokine secretion. So combating against a virus and cancer is more difficult if you take it in evening. So generally, if you see the vaccination, generally the doctors ask us to come on morning for vaccine. So this is the reason. And similarly, for influenza, at 9 to 11 a.m., we find higher antibody response. And if it is taken on evening, that is at 3 to 5 p.m., lower antibody response. Similarly for SARS-CoV-2, so if we take at morning 9 to 11 a.m., so see, I don't know whether you realized all this, but this is the fact, okay? So we find higher neutralizing antibody, and if you are not getting fever at the evening, so we are getting these kind of, we are getting the insignificant number of cells are creating, immune cells are creating from that, okay? So significant number of cells are created on morning. Even if you create a kind of a fever after taking the vaccine, it means that we are more effective. It's going to get more vaccination for at least the prolonged response for few more days. Got it? So stronger B and helper T cell response, higher percentage of monocytes and dendritic cell, higher percentage of memory B cells. Antibody modeling through squid injury. I told you, the antibody is an antibodyomics. So we need a kind of a commercial software. We have a demo. We have a steps to guide each and every step can guide you how to go with the engineering and after modeling how to go with the docking. Everything is integrated in this kind of a package. Antibody modeling can also be done with the DNA star. Again, this is also another commercial software. That's it. So I completed a kind of a computational vaccine design today. Okay, only thing is that I have to take you to the Vaccine server. Okay, so we have to take you to the Vaccine server. I hope I'm in line. Any doubts up to present? So I think I can take up to nearly 10 minutes, not more than that. Okay, so we have to. If you have any doubts, you can ask. Am I audible to everyone? Yes or no? Because I'm continuously taking internet. I don't know whether I'm. Hello, hello. Am I connected to internet?
Yes, sir.
Okay. Okay. Because see, even here it's not vibrating. How about others? Am I audible to everyone? Yes or no? Yes, is it right? I just check it. What happened to my browser? Yes. Yeah. Thank you. Because we don't know. Even I too am bothering about the data. Okay, anything. I have data. No problem. Okay, let me take you to the next vaccine. So, in vaccine, okay, we can violinate is the page, isn't it? So we can go to the violinate initial violinate.org. So I'm taking to this violinate.org. Go to this wax query. First, I have taken to the human vaccine urgent. So I take it to the human vaccines. In human vaccine, how to search uh, any pathogen, bacillus anthracis. Can we check? Search. So we got bacillus anthracis. So if you want to go with anthrax vaccine, this anthrax virus, this I mean anthrax bacteria. Okay, so deadful bacteria. Even this is a bio weapon. I think if you search in the world for this, also used for can be used as a bio weapon. And I think 4 years before, even IIT Madras, the teas, okay, some two varieties of teas got infected with the anthrax. But fortunately, immediately the vaccination team came and just cleared all the debris, and we are free from this kind of thing. When I worked PDF there, we found this kind of thing happened. Okay, but we have to maintain stringent protocols. Okay, for this anthrax vaccine of human type, I just searched. Okay, so more about this information of the anthrax vaccine, we can find this from this website, Qvax. Okay, and related to the veterinary vaccine. Okay, so how to search? So we can search based on now another avian paramoxy virus. This is also often occurring in parts of India. Okay, bird flu, avian paramoxy virus, the chicken. Okay, millions of chickens were sacrificed if this virus is infected. So for this, the vaccine name is this. Okay, USDA number is provided. We have type 1, type 3. So more about this information of this vaccine, we can get this information there. Okay, to check whether the vaccines are available for particular viruses or bacteria, we can able to go through this. Okay, so if I click that, it direct to the onto B. So this is an ontology information of the vaccines. Okay, how these vaccines are developed. For chicken vaccine, we have licensed chicken vaccine. So these are the types of vaccines given to the chicken. In the poultry, one has to check whether these vaccines are given to the poultry or not. Okay, these much vaccines are developed for the chicken. Okay, now let me take you to the next thing. Okay, wax vector. So I close these windows, whatever I opened. Now I take you to the for COVAX we have separately COVID-19 vaccine. So Vax vectors, a database of recombinant vaccine vectors and vector-based vaccines. So for vectors we have separately, and if you want to check what is the vector and how it distinguishes from plasmid. So generally plasmids we take E. coli plasmids because of rapid multiplication, and so only we are using that in recombinant production of human proteins, isn't it? Even the animal proteins can be produced from that. Okay, and if you see such a kind of proteins production from recombinant vectors in the plasmid, we just remove a part of a DNA and we incorporate the corresponding insert plasmid where we want, like if you want to produce recombinant insulin, we insert human insulin DNA in a E. coli plasmid vector, so that the during the multiplication of E. coli cells, we can see the human insulin production is increased, isn't it? Similarly, for producing vaccines in pilot scale, one should have a knowledge on vectors. So one should need what is the plasmid DNA information, and what is the insert DNA we can able to insert. Okay, for vax antigens, blast can give you such a information on what antigen has to be incorporated. Okay, what insert DNA can be incorporated into the plasma. So for vax vector query, so we have to go with for example, if you search for the cancer, Okay, we can find this much number of vectors for the cancer. Okay, so we can choose any of this. So if you see the number of vaccines used in this vector, the maximum they have tried this Salmonella enterica. This is the most widely used vector. Okay, and if you go inside, we can get information on this. Okay, so like this, we can see that information whatever vaccines available in the market. So we can get the references and we get few more information from Violi Net, that is a web-based vaccine design and analyze system. So similarly, we can search vax so that is vaccine adjuvants. I told you now I have developed a vaccine adjuvant database. Whatever vaccine adjuvant database I developed is with a plant-based molecules. The plant-based molecules and that is very limited, up to 1,000 interactions. I just tried with nearly 11 plant molecules, I believe, not more than 11 plant molecules. But this is a major database, a large database. Here they have used types of. So if you see, if I want to search on adjuvants, we can go with it. Okay, so if I want to search, I have to give any keyword is mandatory actually. The keyword, the most important keyword is select the type of vaccine. Okay, the others are the additional filters. Okay, now I search for the live attenuated vaccine. For live attenuated vaccine, we have friends, complete adjuvant and friends, incomplete adjuvant. Other than that, Arlaccal, can we go with Loxapamide or even DHEA vaccine adjuvant? I'm taking it to the DHEA vaccine adjuvant, so we get this information on this. Okay, to get more information on this, go to this and you find this. So actually, this DHEA is a chemical dissolved in 50% ethanol. So whatever dissolved in that 50% of ethanol can be used as a. So this is a chemical. Okay, this is a chemical dissolved in ethanol which can be used as a. Similarly, doxorubicin. Okay, so more about this adjuvants we can find in vaccine adjuvants. So like this we can find for DNA vaccine and we have similar kind of an information. Okay, this is wiring. And, so we can see similarly we can also can able to download the information based on the different cluster of genes pathway. COG is cluster of genes. So can link with the type of pathways. Okay, if you want to locate with respect to subcellular thing, we can also locate to the subcellular information. So we have plenty of information. One day thing is that we have to go through that. The remaining Discovery Studios, Croidinger, DNA Star, these are commercial software to work. Okay, unless we have commercial software, we can't able to go through. So almost I have completed today's session. And tomorrow's session, we have only one session on tomorrow. So on tomorrow's session, we are going to discuss on artificial intelligence and machine learning approaches of drug as well as vaccine design. So of course, tomorrow is the most important session, which is linking all the concepts, whatever we discussed from day 1 to day 13. We have completed 13-day sessions. So tomorrow is the 14th day session. And after that, we are going to have an MCQ and feedback. Okay. Any doubts, you can ask me in person. So tomorrow we have at same time, 7 to 8 o'clock. I think we don't need to change the time. 7 to 8 o'clock, we can have this. Or if you want to prepone, let me know here. Through even through WhatsApp group, if you mention, then I can ask the opinion of the others and I can change. Okay, so tomorrow is the last session. Don't forget. Okay, any questions? You are welcome to ask from any of 1 to 13 sessions. So, if no doubts, thank you very much even participating these sessions, these two sessions on the holiday. Okay, pardon for the trouble, and I thank you for the interaction as well as for maintaining silence during my sessions. Thank you. Have a great day.
Thank you. Thank
Good
you.
night.